To perform PCR amplification, you need to combine all the components required for DNA replication. These are as follows.
Sexing by PCR of female and female DNA's electrophoresis was carried out in 0.8 agar gel with tie buffer. The bands were visualized under ultraviolet transilluminator. Lanes 9 have 1 kilobase plus, lane 10 had 3 microliters of DNA sample, lane 11 had 3 microliters of negative control that was water, and lane 12 had 3 microliters positive control that was PCR products. The agar gel was strained using gel red. It is important to prevent primers designed to form secondary structures or interaction between them. There are three structures: hairpin, homodimers, and heterodimers. For homodimers, it is acceptable a delta G bigger than minus 5 kilocalories per mole, and a delta G bigger than minus 6 kilocalories per mole. Heterodimers formed by intermolecular interactions between the first signs and the antisense chains. It's acceptable a delta G bigger than minus 5 kilocalories per mole and a delta G bigger than minus 6 kilocalories per mole. In the case of hairpins, they are formed by intermolecular interactions which affects the amplification process. It's acceptable a delta G bigger than minus 2 kilocalories per mole, and it's important to break the hairpin structures formed in the 3' extreme. In the case of the primer forward, we have a delta G of minus 0.98. At this energy is required for the breaking the structure. The homotimer has a delta G of minus 5.02, and the heterodimer has a delta G of minus 5.25. In the case of the primer reverse, has a delta G of minus 0.28. It is the energy required to break in the structure. And the homodimer has a delta of minus 549 delta G. In the next chart, we have the specification of the primers. In the first column, we have the forward primer sequence that was 5' to 3'. In the second, we have the reverse sequence primer that is 5' to 3'. And in the third, we have the temperature of melting of each primer. Fourth, we have 58.3 degrees Celsius, and for the primer reverse, we have 55.1 degrees Celsius. We conclude that if they are not very stable and can be uses that don't significantly affect the process because they do not form the secondary structures. In the lane 4, we have a PCR product degradation that is in color pink, and in color green we have PCR product. This is in the band 5000 and 3000. And in the color blue we have the NTPs and primers. This is running more than the marker. We have 0.8 agar gel on lanes, 1 kilobase in vitro gene marker and was 5 microliters, 2 genomic DNA sample 5 microliters plus 2 microliters of loading buffer, 3 negative control that was molecular biology water grade that was 5 microliters plus 2 microliters of loading buffer. 4 we have positive control of PCR products of 447 pairs of base that was 5 microliters plus 2 microliters of loading buffer.
When performing a PCR, one has to take care of several things, including pre-PCR activities, which include protocols and equipment necessary for nucleic acids extraction, sterile and contaminant-free reagents, and working space, as well as good care during sampling are critical for a PCR reaction. Also, when preparing the PCR reaction, the amount of polymerase, DNATPs, buffers, and primers should be added according to the concentration of the sample to be amplified. Before starting the PCR reaction, parameters such as time, temperature, and number of cycles need to be established according to the enzyme used and the primers added. If a PCR cycle is set without considering this, it must likely to fail. Most of the times, an unsuccessful PCR resides in the quality of the sample, which may be affected by the extraction protocol performed, contamination by DNA or salts, among other things. According to Laura Espinosa from the National Institute of Ecology and Climate Change, if you're to perform several PCR reactions, it is highly recommended to use an extraction kit from a reliable vendor. That way, it is mostly guaranteed to have a better outcome. On consequence, we agree that above all steps during the preparation of performance of PCR, it is critical to have excellent pipetting skills in order to avoid contamination and/or loss of samples. Also, it is important to know the working parameters of the enzyme used and the proper concentration of reagents for a PCR reaction. About the results obtained in this practice, the most significant would be the fact that the DNA sample is not observed on line 2. We attribute this event to the fact that, during DNA preparations, the sample was quite diluted with water, and maybe that's why there is no PCR product. It is remarkable that, as expected, on lane 3 there is no sign of DNA contamination, therefore it can be assumed that the protocol was successfully followed. As for lane 4, as expected, the PCR product is observed, with little degradation and a possible cell contamination. At the very bottom of the agarose gel, in lanes 2 to 4, it can be observed 2 marks, which we attribute to the existence of unused DNTPs and primers during the PCR reaction. This may be possibly due to the high concentration of DNTPs, but this is not a determinant factor for the results, because if more DNTPs are added, this may kidnap magnesium and inactivate the polymerase. Therefore, the zero obtention of DNA on lane 2, but the same would have happened on lane 4, but it didn't.
Polymerase chain reaction (PCR) is one of the most used methods for amplification of sequence of interest. In this work, such method was used to a genetic easy determination of gender, including a co-amplification of zeta if X and zeta of J target. In order to obtain a successful outcome, it is necessary to have an appropriate procedure, which starts from the pipetting skills, preparation of the reagents, as well as sample and the correct handling of these. It is important to remark that the extraction of nucleic acid was a critical point at which contamination can occur with proteins, DNases, enzymes which are responsible for the degradation of genetic material, and other reagents that can alter the results and therefore report basis conclusion. Also, when performing PCR, number of setting cycles of extremely important because the change of temperature and excess of time can lead to the inactivation of the polymerase. Lack of PCR reagent or even amplification of unspecific reagents, thus drawing basis results.